You know, to me, it is really cool that cells are the structural unit of all living things. Cells are the functional units of all living things. Cells carry out a multitude of chemical reactions inside their cytoplasm and on the surface of their membranes. But even beyond that, it is just ultimately cool, indeed, far out, that cells communicate with each other. So this is accomplished by three basic mechanisms, one of which is called a gap junction. A gap junction is actually a pore that extends from one cell into another, so chemicals that are in the cytoplasm of one cell can be shared with the next cell. So it is very common for cells to have gap junctions. When we were talking about cardiac muscle tissue, I mentioned those things called intercalated disc. The intercalated disc is actually a gap junction through which ions such as sodium can pass from one cell to another. When we get to our cardiology chapter, you will see how necessary that is, that one cell be able to communicate with the other in that way. So many cells have gap junctions and they can allow certain things, ions or nutrients, to pass from one cell to another. So that's one mechanism by which cells can communicate with each other. A second mechanism is by way of certain secretions that the cell gives out. If the secretions go into the blood and have receptor sites at distant cells, then we call those secretions hormones. There are other secretions which do not go into the blood, but they have receptor sites on the nearby cells, cells that are in the same tissue. Those are called paracrine secretions. And then there are others that are simply called signaling molecules. So what's going on here is that the cell, by the secretions that it produces, is communicating with other cells, and those cells communicate back to it. The third mechanism by which cells can communicate with other cells is by way of nervous connection. So in the nervous tissue and physiology section of this course, this is what we're talking about, is communication cell to cell by means of the functional units, which are called neurons, and the chemicals they release, which are called neurotransmitters. So let's have a close-up look at nervous system physiology. I think you will enjoy it. Next slide, please. You know, it may seem like we never get out of histology in this class. Well, there's a reason for that. I think I told you in the beginning that I don't think a student really understands anatomy unless they understand it at the microscopic level. So these three images should look familiar to you. We covered them in the histology section. So these are neurons, nerve cells. Nerve cells are very large. They oftentimes will have extensions from them that may actually be measured in feet as far as their length goes. So when you take a histological section to show nerve cells, well, obviously you're not going to be able to see the whole thing because they may be 1, 2, perhaps even 3 feet long, all said and done. That long extension from the neuron is the axon, so that is what is most likely to get cut off. Looking at the image on the left, you should be familiar with these cells. We just talked about them and their function in the motor cortex. These are the pyramidal cells that issue the notion to make a motion insofar as we are able to understand how the will of a person actually fires an action potential that results in muscle contraction. Well, from all that we know, these pyramidal cells are where that begins. So that is a histological section of the motor cortex showing those pyramidal cells. Anytime you look at nervous tissue under the microscope, you will see 1,000 to 1 other types of cells. Well, those are called glial cells. Glia means glue, which kind of suggests that they hold things together. Well, they have a lot more functions than that, which we will talk about later. In the middle image, that is a typical spinal cord neuron. You can see how large the soma is. You can see the nucleus, and usually you can see the nucleolus within the nucleus. So also surrounding this particular neuron, you can see many glial cells. In the image on the right, that is a section taken from the cerebellum, those unique looking neurons there that kind of line up between the granule layer and the molecular layer, those are called Purkinje cells. They are actually in the sensory pathway. So when we are sending signals toward the central nervous system, then those are sensory signals, and these Purkinje cells are in the sensory pathway. We devote a whole lot of our body's attention to coordinating movements and maintaining balance. The cerebellum has many functions, but that is probably chief among the functions of the cerebellum is simply coordinating movements and maintaining our balance. Remember that we walk on 2 feet. That is not ideal for maintaining balance. It's easy to fall down if you only walk on 2 feet. How often do you encounter a table with 2 legs? Or what if you sat down on a chair that only had 2 legs? Well, you're liable to take a tumble. Anyway, back to the point here, the Purkinje cells, you can see them, and then surrounding them, you can see the heavenly host of glial cells that are there to support and to nourish the actual neurons. So that's how our histology of the nervous system looked when we went through that. So I wanted to just briefly revisit that. Next slide, please. Well, this is a pretty nifty illustration of how the neurons and the supporting glial cells kind of relate to each other. The large yellow cells there are the neurons. You can see the nucleus colored somewhat orangish pink, and within that you can actually see the nucleolus. The many extensions off of the neurons are called dendrites. They receive incoming signals. The long extensions that come off of the neurons are called axons. And if there is an action potential to be fired, then it would be fired down the length of that axon. So we have several receiving areas. The nerve cell body itself, which is called the soma, has receptors. The dendrites certainly have receptors, and even some axons have receptors for incoming stimuli. But for the most part, we're talking about signals coming in by way of the dendrites or by way of the soma. The supporting cells, the glial cells, which outnumber the neurons 1,000 to 1, they are of 4 different types in the central nervous system and 2 different types in the peripheral nervous system. So some of them are shown here, the central nervous system glial cells, particularly the astrocyte, so called because someone said, well, you know, that thing looks like a star. Well, perhaps. Okay, so these are glial cells. They are not neurons. They don't have the physiologic abilities of a neuron. They are there to support the neurons. So the astrocyte in particular has a great number of functions that it performs, which I will detail later. I think what is trying to be communicated in this particular illustration is the formation of the blood-brain barrier between the capillary and the neuron. Nerve cells are extremely particular about their chemical environment. So just any old thing that might end up in the blood is not necessarily allowed in the extracellular fluid around the neurons. So we call that the blood-brain barrier. There are certain things that are in the blood that we do not want to come into contact with our neurons. The astrocytes are the largest contributors to that. Notice that they have extensions which wrap around the capillary. Those are called perivascular feet, and they are there to prevent certain things from the blood from getting into the extracellular fluid surrounding the neurons. So that's one of the functions of the astrocytes. There are some others that we will talk about later. Now look at the glial cells that are called oligodendrocyte. Oligo means a few, dender means branches, so these are cells that have a few branches. Well, that's fine. In the brain and central nervous system, the oligodendrocytes form what is called the myelin sheath around the axons of our nerve cells. So we have myelinated axons. We call that later white matter. The reason for that is because myelin is white. We also have non-myelinated axons. In that case, those would end up being called gray matter because they do not have myelin. So it is the oligodendrocyte itself. It isn't so much a secretion of the oligodendrocyte, but the oligodendrocyte itself wraps around the axons, forming the myelin sheath around those axons. If an axon is myelinated, it will transmit an action potential much faster than a non-myelinated axon. Well, it turns out that we have a need for both white matter and gray matter, so it isn't necessary for all nervous signals to travel at such a high rate of speed. In some cases, it is necessary, so we do myelinate those, but we don't myelinate those where the speed of transmission is not such a big deal. Also, we have down there in the southeast corner and at the eastern side of the view, the microglia. The microglia are actually modified macrophages. Macrophages are cells of our immune system whose job is to find anything that needs to be eaten and destroyed and eat it and destroy it. So if we have debris or foreign things such as viruses, bacteria, protozoa, fungi, anything, or worn out cells that need to be digested and disposed of, then that's what the microglia do. That's the same thing that macrophages do in the rest of the body. These are specialized macrophages designed to be the garbage collectors in the cerebrospinal fluid. So the microglia are phagocytes. They eat, digest, and destroy whatever needs to be eaten, digested, and destroyed. Remember, the neurons are very particular about their environment. We cannot have foreign things or things that would clutter up our cerebrospinal fluid just floating around. So the microglia are there for that disposal type of function. A fourth type of glial cell also is shown here, the ependymal cells. Notice that those are little box-shaped epithelial cells. Well, we call those, we would call that a simple cuboidal epithelium. When we talked about epithelial tissues, I mentioned that one of the functions of epithelial cells is that they secrete things. Well, it turns out that the ependymal cells, which are particularly found in the walls of the ventricles of the brain, they secrete the substance that we call cerebrospinal fluid. So the neurons themselves are surrounded by cerebrospinal fluid, which is taken from the blood but processed by the ependymal cells so that it's appropriate for the environment that the neurons need. Neurons are very particular about their chemical environment. So cerebrospinal fluid is not the same as just any other extracellular fluid. It is not the same as blood plasma. It may be very similar to those things, but it is processed by the ependymal cells and released into the cerebrospinal fluid so that the cerebrospinal fluid has a particular balance of electrolytes and other chemical compounds which are appropriate for the nerve cells. Okay, so that is a composite look at the neurons and the glial cells associated with them, and we will detail that a little bit more in a later slide. Next one, please. Well, by now you should be an expert in the subject of polarized membranes. We have spent some time talking about muscle tissue and how muscle cells maintain a polarized membrane. Maintaining a polarized membrane requires the expenditure of energy, even though we have more negative entities inside the cell. That would be the phosphate groups, the amino acids, and the nucleic acids. We have to drive that polarization even farther. In the case of muscle cells, we use the sodium-potassium pump to maintain a resting membrane potential of about minus 90. That means that it was 90 points more negative on one side than on the other. So that is a trigger waiting to be pulled because we have a separation of charges, and nature hates a separation of charges. Nature is always inclined toward equilibrium. So when we have a separation of charges, then we have a trigger waiting to be pulled. We would call that excitability. So here we say, well, characteristics of nervous tissue. Well, nerve cells maintain a polarized membrane. So they are excitable. They are sitting and waiting for the trigger to be pulled so they can fire something. What they're going to fire, we will call an action potential or a wave of depolarization. So the resting membrane potential for most neurons is at about minus 70 millivolts. That means it is 70 points more negative on the inside than it is on the outside. Okay, so excitability we have talked about with muscle tissue and nervous tissue. We did not mention anything like that associated with epithelial cells or connective tissues. So it is unique to muscular and nervous tissue. Also, the ability to transmit the signal. So we might have a depolarization by the opening of sodium channels. We might get depolarization at a particular location, but how does that translate into the transmission of the depolarization from point A to point B? Well, that's called conductivity. Muscles do that. Nerve cells also do that. So in the nervous physiology, we will call that an action potential. So if we get a threshold stimulus, that is a stimulus of sufficient intensity to initiate a wave of depolarization, then we might possibly fire an action potential, which is the transmission of the signal to the next neuron or to the target tissue. Okay, secretory function. Well, this is interesting. Neurons, in addition to being excitable, in addition to having this conductivity, neurons are known to secrete things. Well, we do know that they secrete chemicals that transmit the action potential to the next neuron or to the target tissue, so we just call those neurotransmitters. But there are many neurons, which are called neuroendocrine cells, which release chemicals into the blood. Well, when we release chemicals into the blood that are then going to bind to distant cells, we call those hormones. So the hypothalamus, which is part of the brain, well, the brain has nerve cells, neurons. The hypothalamus alone makes 9 different hormones. The adrenal medulla releases into the blood a hormone called epinephrine. Those cells in the adrenal medulla are actually neurons. So we have neuroendocrine cells. They are both neurons and secretors of hormones and paracrine signaling molecules. The last characteristic that I've listed here is the non-mitotic nature. When we are born, we have all the neurons that we're going to get. Those neurons are designed to stay with us lifelong, so they are unusually long-lived cells. But the drawback there, since we don't have stem cells to replace them that are lost, they don't undergo mitosis. Any that are lost due to disease or injury, you simply have to live without. Well, this opens up a pretty interesting area of biology these days, which is totipotent stem cell research or embryonic stem cell research. Those particular cells are totipotent because they can differentiate into any of the cell types that we have, including neurons. So even though we don't have in our bodies stem cells to replace lost neurons, it is possible that we could have them by way of taking embryonic stem cells from a developing embryo or from a clone and using them to replace brain tissue, spinal cord tissue, peripheral nerve tissue, which has been lost due to disease or injury. So that type of technology is somewhat available now, but the wrinkles still have to be worked out. So the characteristic here, it is just a general rule that nerve cells do not replace themselves. If they are lost, then you have just simply lost it. Now, that does not mean that a nerve cell that is injured cannot heal. Remember, nerve cells are very long. It is the soma of the nerve cell that must be destroyed in order for it to be lost. If an axon is cut off, an axon can grow back. So that doesn't involve mitosis. That's just like a salamander having its leg cut off and it grows a new leg. If you wanted to kill the salamander, well, you would have to kill the salamander's body. You can't do it by just cutting its leg off. So the axon can regenerate. So you can have an injury to a nerve. A nerve is a collection of axons. It's only if the nerve cell body is destroyed that the nerve cell itself is gone for good. If the axon is injured, it can regenerate. Well, I kind of stumbled around on that last part, but I think you got the point. Next slide, please. Well, there's no such thing as a perfect illustration of the anatomy of a neuron. And the reason for that is because we have neurons that come in a variety of shapes. Some of them are called unipolar. They extend out only in one direction. Some of them are called bipolar. They have extensions going in two directions. Some are called multipolar, such as the one you see here. You've got one long axon going this way, but you have multiple dendrites at the other end. And then there are some neurons which are anaxonic. They don't even have an axon. I mentioned earlier the nerve cells that are in the adrenal medulla. Those are neuroendocrine cells. So rather than firing an action potential down their axon, they don't have an axon. So what is their function? They secrete epinephrine. So they're neuroendocrine cells. They are actually nerve cells which have a hormone releasing function rather than transmission of action potentials. So all that said and done, this particular illustration is very useful. It shows a multipolar neuron, which is quite typical of many. Remember that the cell body at UCA, we would call the cell body of a nerve cell the soma. Inside that, we have a large nucleus, usually a visible nucleolus. The endoplasmic reticulum of nerve cells is compartmentalized into little box-shaped groups. It has the same function as any other endoplasmic reticulum, which is largely the modification of polypeptides synthesized by the ribosomes. The compartmentalized endoplasmic reticulum of neurons is usually called Nissl substance. That's Nissl. It's a person's name. Nissl substance is just the compartmentalized endoplasmic reticulum of nerve cells. Nerve cells are highly metabolic. Anything that is highly metabolic has to be well equipped with mitochondria. It is the mitochondria that makes ATP, and ATP is what drives metabolic activity. So nerve cells are well equipped with mitochondria. There are also the other neurons, the other organelles present in neurons that you would find in any cell, such as the Golgi apparatus, peroxisomes, lysosomes, and things of that sort. Since I mentioned lysosomes, let's play with something here. It just came to my mind, so let me throw it out at you because it becomes important. The lysosomes are little vesicles of digestive enzymes which break down, that's what digestion is, break down things which might get in the cytoplasm. They can be digested and destroyed by the digestive enzymes of the lysosomes. So that is called hydrolysis. The lysosomes use water plus the enzymes they have in storage to digest and break down things such as debris or junk which might get inside the neuron cytoplasm. Well, remember that neurons are lifelong. It's not like you're going to get new neurons every year. The same neurons you had when you were born are the neurons that you will have the day that you die. Well, that means a whole lot of junk has to be digested and disposed of over the number of years that a person lives. That could turn out to be a lot. What if the lysosomes are unable to digest and destroy those things? Well, it ends up accumulating in the cytoplasm. So neurons are notorious for having those types of accumulations in the cytoplasm. One of them is called lipofuscin. Lipofuscin accumulates in the cytoplasm of nerve cells over years, and other things might also. Well, what if your car's engine has metal shavings circulating around in the oil? Isn't that going to eventually interfere with the functioning of your car's engine? Well, see if you can make the relationship there. Things that accumulate in the cytoplasm of the nerve cell eventually will interfere with the functioning of the nerve cell. The nerve cell is lifelong, so it has many years for things to accumulate. A lot of junk can accumulate, and at some point that junk may get in the way of the functioning of the nerve cell. Well, what if that nerve cell controls your movement? Then you're not going to move very well. What if that nerve cell is involved in your cognition? Then you're not going to think very well. What if that nerve cell is in your visual pathway? Then you're not going to see very well. So it is one of the many explanations for senescence, why people lose certain functions as they age. I'm not sure just what got me off on that, but let's move on. The area there called the axon hillock. Inside the axon hillock, we have what is called the trigger zone. Remember, I referred to a polarized membrane as a trigger waiting to be pulled. Well, the axon hillock has the trigger zone. That's where the stimulus has to register threshold or better. A minimal stimulus will not pull the trigger. A threshold stimulus will pull the trigger, so it's called a trigger zone. So the wave of depolarization, if it is of sufficient intensity, will pull the trigger at the axon hillock, and the wave of depolarization then progresses down the length of the axon. All right, getting to the end of the axon, what is called telodendria there, that's fine. Telodendria just means terminal branches. The synaptic terminals, there are little dilated sacs at the synaptic terminals that we call the synaptic knobs. It is within the synaptic knobs that we have the synaptic vesicles, and the space between the synaptic knob and the next neuron or the target tissue is called the synaptic cleft. So neuronal function is electrical and chemical. So that was not known until the 1800s. A scientist named Raymond Cajal, C-A-J-A-L, Raymond Cajal, discovered this, that there is no physical connection between neurons. There is a small gap, and so we call that gap the synaptic cleft, and the bridging of the gap is accomplished by the chemical released, and that chemical released is called the neurotransmitter. So we have more than 100 chemical compounds that we call neurotransmitters, and we'll talk about them another day. Next slide, please. Our nervous system is a two-way highway. So we have signals that are traveling from the body's tissues toward the central nervous system. That would be the brain and spinal cord. So those signals are called sensory or afferent signals. The other side of the highway has signals going the opposite direction, so they're traveling from the brain toward the body's tissues, so we call those efferent or motor signals. The motor signal may be voluntary, such as your notion to make a motion, something you decide to do, Well, that stimulus begins in the motor cortex with those little cells that we call the pyramidal cells. If the motor function is autonomic, it is not something you choose to do, it is something that is done autonomically. Those stimuli generally originate in the hypothalamus of the brain. There are no lights on in the hypothalamus, so we don't do things voluntarily. They are done involuntarily, such as the constriction of the arteries to raise the blood pressure, or the movement of the intestinal walls in the process of digesting food. So those are motor pathways. What about the sensory pathways? Well, they are going from the body's tissues toward the brain, and we have specialized collections of cells called receptors. Receptors are where the action potential is initiated if it is a sensory function, an afferent function going toward the brain. So it's kind of difficult to define a stimulus because we're really trying to identify, well, where does this thing start? Well, the best that we can understand is that the voluntary activities of the body begin with the motor cortex, and the involuntary motor functions originate in the hypothalamus or somewhere thereabout. The sensory stimuli originate with these things called receptors. Rods and cones in the retina of the eye are receptors. That's where the signal originates. It then is propagated by way of nervous tissue toward the occipital lobes of the brain, where it is then perceived and interpreted. So defining a stimulus is kind of like saying, well, where did this thing begin? So we know then that a stimulus causes or is the manifestation of an influx of sodium into the cell so that the membrane depolarizes. Well, that's what we're talking about, whether it be light shining on the retina or someone poking a pencil in your leg. It's a stimulus. That's where it starts, and it's sent to the brain, or it's sent from the brain to the tissues. So when we have a stimulus that causes sodium channels to open, therefore depolarization at a certain location on a nerve cell, for instance, a dendrite. When those sodium channels open, we need to know, well, how much sodium comes in? We maintain a resting membrane potential at minus 70. The question is, how far down or how far towards zero do we need to go in order for the trigger to be pulled? Well, that would be called the threshold. So if we are setting at minus 70, maintaining that as our resting membrane potential, what we have found out is that in order for the stimulus to be propagated, to go down the length of the axon and to be transferred to the next neuron or target tissue, we need to bring about minus 55 to the trigger. So we were at minus 70. The stimulus needs to be of sufficient intensity to bring a minus 55 to the trigger zone. Now, we have to consider the summation of stimuli. A nerve cell may receive signals from thousands of other nerve cells. So we're going to have to summate what is the sum of the stimuli that are arriving at the nerve cell. Well, another thing that we have to consider is that some stimuli don't depolarize, they in fact hyperpolarize the receiving nerve cell. So we can have a stimulus which causes sodium channels to open. That's going to depolarize, but then there are also stimuli that can open chloride channels. If chloride channels are open, chloride is a negatively charged ion, then it will contradict the signal or the depolarization that is offered by the sodium. So we have to summate. We have to take the sum of all incoming stimuli, and when all is summed up, have we delivered to the trigger at least a minus 55? Anything greater than 55, it doesn't matter. If we pull the trigger, the bullet fires. It doesn't matter if you pull the trigger harder. If you pull the trigger harder, the bullet doesn't fly any different than it would if you just pulled the trigger enough to get it to fire. So that's the way the neuron operates. If we can bring a depolarization, that is going towards zero from minus 70. So from minus 70, by the time we get to the trigger zone, we need to still have at least a minus 55. So that's a depolarization of 15 points. That would be threshold. That is sufficient to pull the trigger. When the trigger is pulled, the action potential is fired. Doesn't matter if you pull the trigger harder, the bullet doesn't fly any faster. The action potential is going to travel at the same rate down the axon, no matter whether you brought a minus 55 or whether you brought a 0. So getting to threshold is a function of summation. If we have depolarization due to sodium influx, but we also have hyperpolarization due to chloride influx, then we have to summate the two. If the sum of the two brings us to a minus 55 or better, then we can pull the trigger and the action potential is propagated down the length of the axon to the synaptic knobs. If the action potential reaches the synaptic knobs, calcium enters into the synaptic knobs and the neurotransmitter is released. Thank you very much. Well, this is just a funky little illustration that I came up with using the tools available to me on the PowerPoint. So I've got these lines and I've got this oval. Well, see if you can use your imagination just a little bit and see that green egg-shaped thing. Well, that is the soma of a neuron. Over to the western side, I've got 4 blue lines drawn there. Those are the dendrites. And going toward the east coast there, I've got that long extension. Well, that would be the axon, and the little branches off of it are the terminal arborizations leading to the synaptic knobs. So that's what the illustration is all about. Now let me show you what the numbers are all about. So I'm talking about the difference between a local potential and an action potential. A local potential is an area of depolarization in the nerve cell body. I told you on the previous slide that it is a summation of stimuli. Some stimuli are depolarizing, influx of sodium. Some are hyperpolarizing, influx of chloride. Chloride is negatively charged. Sodium is positively charged. So look at the 4 stimuli arriving at the dendrites in this little illustration. So we have a dendrite that is bringing in a +5 depolarization. Remember, the membrane potential is at -70. We need to deliver a -55 or better to the trigger in order for the trigger to be pulled. But we have to summate, so let's take into account everything that's coming in to the neuron. So the first one, that's a +5. That means sodium channels open, and our change is 5 millivolts from the -70. The next one down there also is a +5, so that brings us to 10 against a -70. The next dendrite is bringing in a hyperpolarizing stimulus. Chloride channels are opened, so we go -5. So far we have +10, -5. The last little dendrite down there also is a depolarizing stimulus, so that adds +5 to our number. So what we have is +5, that's 10, -5, well, that brings us back to 5, +5 equals 10. So the summation of stimuli arriving at the nerve cell body comes to +10. Our resting membrane potential is -70. The summation of incoming stimuli amounts to 10. So -70 plus 10 equals a change of, or a change to -60. That is not sufficient. So we call that a local potential. There was a depolarization that occurred, but it was not sufficient to pull the trigger. We got to -60. If we don't get to -55 or better, then threshold is not reached and the trigger is not pulled. So local potentials are decremental. They will fade with distance. So if we bring a depolarization to the dendritic end of the neuron, it will fade before it reaches the trigger zone. So it's going to lose some of its punch, in other words. They do not reach threshold, and in fact, they are reversible. Reversible meaning we can even hyperpolarize by allowing more chloride in than we allowed sodium in. Okay, so in this particular illustration, we have a local potential of +10. A +10 is going to fade before it reaches the trigger zone, so it loses some of its punch. So by the time that local potential reaches the trigger, it is nowhere near -55, so it is not going to fire an action potential. Now, put this in real life terms. Let's say that there are bacteria on your skin. Well, you have touch receptors in your skin. Why do you not feel the bacteria on your skin? Your skin is actually covered with bacteria, but you don't feel them. The reason you don't feel them is because no action potential is fired. They are there. They have mass and occupy space. Some of them move about, but you don't feel the bacteria on your skin because the stimulus is sub-threshold. You don't feel it. There are wavelengths of light which your eyes don't see. You don't see ultraviolet light. You don't see X-rays. The reason for that is because they don't fire an action potential. So only those things that bring a -55 or better to the trigger zone is going to be propagated and therefore reach the target tissue. So there are many sounds that are made that you don't hear. That doesn't mean the sound wasn't made, it was made. It's just that the stimulus did not reach threshold, therefore the signal did not reach your brain, so consequently you didn't hear it, even though there certainly was a sound. Okay, so in order for the signal to be transmitted from the receiving neuron to the next neuron or to the target tissue, we have to pull the trigger. Pulling the trigger is dependent upon the summation of stimuli. Some stimuli are depolarizing, they open sodium channels. Some are hyperpolarizing, they open chloride channels. So that is a local potential. Now let's look at the next illustration for an action potential. Okay, in this particular example, the incoming stimuli, we have a plus 10, plus 10, plus 10. So that is sodium gates opening up, so the depolarization due to those incoming stimuli is plus 30. There is then one hyperpolarizing stimulus coming in that is a minus 5. So we summate. So 30 minus 5 gives us 25. Our resting membrane potential is minus 70. We need to bring a minus 55 or better to the trigger zone in order for the trigger to be pulled. So we have a local potential of plus 25. As it travels through the nerve cell body, advancing upon the trigger zone, a local potential of plus 25 would probably fade some. We might lose some of our punch by the time we got to the trigger zone, but a plus 25 against 70 brings us to minus 45. Well, we needed a minus 55 to pull the trigger, so even if it does fade a little bit, it still should bring a threshold stimulus to the trigger. So if we get a minus 55 or better, which in this case, if there is no fading out of the signal, we brought a 45, so the trigger is going to be pulled. So that's why I have the red arrows going down the length of the axon. The action potential is fired. So we're going to travel down the length of the axon to the terminal arborizations, to the synaptic knobs. Calcium channels open in the synaptic knobs. The neurotransmitter is released. The neurotransmitter binds to the next neuron or to the target tissue. Consequently, the signal is transmitted from one cell to another. So this is an example of firing an action potential as opposed to the previous slide where the stimulus was sub-threshold and therefore no action potential was fired at all. So thankful that we do have a threshold that has to be reached. Otherwise, you would hear every sound made. That would drive you nuts. You would see everything. That would drive you nuts. You would feel everything, taste everything, smell everything. Well, this is why it's good that we have a threshold that has to be reached before a signal is sent. Otherwise, every stimulus uh, would be uh, would be impacting upon your brain, or every thought that you have, you would be acting out with your arms and legs. Imagine what that would look like. So this is how the nervous system works. Signals that bring to the trigger a threshold stimulus will result in an action potential being fired, and so the signal goes in the sensory pathway to the brain, or if it's in the motor pathway from the brain to the body tissues. Well, there is a third possibility. So this illustration here shows you that third possibility. We have four stimuli arriving by way of the dendrites and causing a change in the membrane potential of the nerve cell body. Notice that we have minus 20. That means chloride channels were opened by that stimulus. The next one also, minus 20. That means chloride channels were opened. That hyperpolarizes our membrane. We have a stimulus coming in which is depolarizing. That plus 10 is depolarizing. Then we have another stimulus coming in, which is minus 20. So all said and done, we have a resting membrane potential of minus 70. That's where we keep it. The incoming stimuli, that is minus 60 plus 10. Minus 60 plus 10 equals minus 50. So we did not depolarize anything. We hyperpolarized the cell membrane. So our resting membrane potential is minus 70. The stimuli that came in resulted in a summation of minus 50. So that takes us farther away from zero. So in order to fire an action potential, we have to go towards zero from minus 70 to minus 55. In this case, we went away. So we go minus 70 plus minus 50 equals minus 120. So the cell becomes hyperpolarized as a result of the chloride channels opening more so than the sodium channels opening. So no action potential is going to be fired. So this is called an inhibitory postsynaptic potential. So you might be curious, why would we want anything like this, something that inhibits the transmission of a nervous signal? Well, here's a good example, I think. There are some pain signals which are unbearable. There are people who have taken horrible injuries, and when they arrive at the emergency room, the doc says, "Well, how do you feel?" And they say, "Well, I actually feel okay." Well, you just got shot in the chest. How could you feel okay? Well, I don't know, man. It really doesn't hurt that much. Well, this is an example of inhibitory postsynaptic potentials. We can hyperpolarize some of the neurons in the pathway to diminish the intensity of the signal. If you have a pain signal, which would be unbearable, then we have ways of kind of putting out the fire. So the person may have gotten his leg chopped off, but he says, well, actually, it really doesn't hurt that bad. I have had bee stings that hurt worse. Or the person who took the bullet wound to the chest. He said, well, it feels like somebody punched me in the chest, but other than that, I really am not hurting that bad. The reason for that is because we have along the way inhibitory postsynaptic potentials, which diminish the intensity of the incoming signal. So what should be tormenting or intractable pain, the fire is actually kind of put out along the way by these chloride channels opening up in certain neurons. So this is an example of an inhibitory postsynaptic potential. Make sure you explain that in great detail to little sister. Well, you have seen this before because it's almost identical to what we used as an illustration. The transmission of a motor signal from a synaptic knob to skeletal muscle. Well, in this case, we're not talking about transmitting a signal to muscle. We're transmitting a signal from one neuron to the next neuron. Well, the dynamic is the same. So we have a wave of depolarization, an action potential, arriving at the synaptic knob. When that happens, calcium fluxes into the synaptic knob, resulting in the release of the neurotransmitter into the synaptic cleft. When the neurotransmitter is in the synaptic cleft, it can bind to the receptors on the next neuron. The next neuron is called the postsynaptic neuron, and it has receptors for certain neurotransmitters. Now remember that some neurotransmitters, when they bind to their receptor, cause sodium channels to open. Well, that would be a depolarizing signal. Others, other neurotransmitters, when they bind to their receptor, they cause chloride channels to open. Well, those would be hyperpolarizing. So we have to take the summation of the two in order to understand whether the signal will be propagated beyond that point. It may very well be stopped at that point. If the hyperpolarization is greater than the depolarization, then the signal is not going to advance beyond that point. Okay, so this is how things work at the synaptic cleft. Now, the only reason that I even brought this up, I think you already understand all of this. I wanted to mention to you that this is probably the most intense area of pharmacological research that we have these days. Since the transmission of nervous signals is chemical dependent, there are about 100 different neurotransmitters. Well, we know what they are. They have receptors. We have some understanding of the activities of receptors. Since it is a chemical dynamic, then it can be manipulated chemically. So we have people who might have certain disorders. They have too little of a neurotransmitter, or they have too much of a neurotransmitter, or they have enzymes that destroy their neurotransmitter or enzymes that enhance the neurotransmitter. So the chemical activity at the synaptic cleft, since it is a chemical thing, that means it can be modified or changed chemically. So we give to people medications. Perhaps you have heard of selective serotonin reuptake inhibitors. Well, the most common or most popular of those medications, I guess, was Prozac or perhaps Paxil. They work at the synaptic cleft. When a neurotransmitter is released, it has to be immediately degraded. Otherwise, it would continue to stimulate the postsynaptic cell. So it is released, it binds to its receptor, it has its effect, and then it has to be immediately taken back up into the presynaptic cell or destroyed by an enzyme or absorbed by astrocytes. Astrocytes do that sort of work. So the length of time that the neurotransmitter is allowed to have its effect can be changed. So the selective serotonin reuptake inhibitor inhibits the reuptake of serotonin into the presynaptic cell, therefore prolonging the period of time that it has its effect on the postsynaptic cell. So this is how something like serotonin or Paxil, antidepressants that were very commonly prescribed, especially in the 1990s, this is how they act. So we manipulate chemically activities at the synaptic cleft, and then we observe the results. Well, the people who were experimented upon taking these medications, if they had uh, problems with mood or depression in particular, many of them reported that they got better. Well, when that happens, then you have a product that you can sell because there are many people who have problems with depression or mood swings or anxiety. And so we manipulate the chemistry of the brain by taking these medications and hopefully getting good results. One other example of this I can give you, uh, I grow peaches, and I have a problem oftentimes with bugs. So I got tired of the bugs. I grow peaches and apples, and bugs, will get on your fruit, and then pretty soon you've got fruit that you can't eat. Well, I got sick and tired of that going on, so I went and bought from Home Depot some malathion. Well, malathion is designed to kill insects, and the way that it works is by inhibiting the enzyme that breaks down acetylcholine. Well, you remember acetylcholine, that's the neurotransmitter that stimulates skeletal muscle. Well, we have an enzyme to break down acetylcholine so that it doesn't continue to exert its effect. Remember that the neurotransmitter needs to be released, it needs to bind to the receptor, it needs to have its effect, and then it needs to be either taken back up into the presynaptic neuron or destroyed by an enzyme or absorbed by astrocytes. Something has to be done with it. Well, the agent that I'm describing, malathion, inhibits the activities of the enzyme acetylcholinesterase. Acetylcholinesterase breaks down acetylcholine. Well, the malathion inhibits the acetylcholinesterase. So what happens? The acetylcholine stays very dense at the synaptic cleft. So if you watch the insect that you spray the malathion on, what you notice is that their muscles go into spastic contraction. That's because the acetylcholine continues to stimulate the skeletal muscle because you have inhibited the enzyme that breaks it down. So malathion is an acetylcholinesterase inhibitor, and the results of that you can see yourself. Just get yourself some malathion, find a wasp, spray the malathion on the wasp, and watch how he starts dancing. The wasp will dance around, jerk around, jump around, and eventually the wasp will die because you inhibit the enzyme that breaks down the neurotransmitter. Well, I went to a great length to tell you that because I believe in giving real life examples. Uh, I guess I need to finish off by saying that I stopped using the malathion, even though it was very effective at controlling the bug population on my fruit trees. Malathion is potently toxic to mammals and birds. Well, I used the malathion and it took good care of the insects. Came out the next day and I found one dead squirrel and two dead birds. Well, that's what they get for trying to eat my peaches and apples. But then I started thinking, I've got two puppies, and it doesn't matter if I give them fresh water. If there's a mud hole in the yard, they just go drink out of the mud hole. So I started thinking, I don't want this malathion next time it rains to end up in a mud hole somewhere, and my dogs go and drink from that. So I had to stop using it while I still had my puppies. So anyway, make sure you tell little sister about that. I hope you can make it more brief than I did, but I am known to go into outrageous detail. All right, next slide. Well, on the very first slide that you looked at, we had four of the glial cells or neuroglia illustrated for you, and I talked a little bit about what some of them do. Here are the six total. The last two are in the peripheral nervous system only. Well, let's start with the astrocytes, supporting or offering a supportive network for the neurons. In other words, giving to them somewhat of a skeleton so that they have a particular location to be and a particular shape to maintain. So astrocytes kind of serve as a skeleton for the nervous system. Those perivascular feet are extensions of astrocytes that wrap around the capillaries to prevent certain things that might be in the blood from getting into the cerebrospinal fluid. I give you an example of that. I had a patient with Parkinson's disease. Parkinson's disease is thought to be due to a deficiency of the neurotransmitter dopamine. And so the recommended treatment then would be, well, give the guy some dopamine. Well, it turns out that there are two stereoisomers of dopamine. There is L-dopa and then there is dextrodopa. So those are stereoisomers. One, they are the same molecule. They are just mirror images. So if that's what a stereoisomer is, it's the same molecule, it's just the mirror image. Turns out the blood-brain barrier can differentiate between stereoisomers. So if you have a Parkinson's patient and the person needs dopamine or would benefit from dopamine, and you give the person dextrodopa, the astrocytes don't allow it to leave the blood and enter the cerebrospinal fluid. In order to cross the blood-brain barrier, the dopamine has to be L-dopa, levodopa, levo meaning left, dexter meaning right. So those are just mirror images of each other, but the blood-brain barrier can differentiate between stereoisomers. So in order to successfully remedy a situation like Parkinson's disease by way of administration of dopamine, it has to be L-dopa. Only L-dopa will cross the blood-brain barrier. The dextrodopa doesn't make it. The astrocytes don't allow it. So it's an interesting example of how the blood-brain barrier works. Okay, converting glucose to lactate for neuron nutrition or nourishment, that is a function of astrocytes. Remember I said on the previous slide that we have neurotransmitters, which once they have bound to their receptors, you gotta do something with them. So they either have to be reabsorbed into the presynaptic neuron, they have to be degraded by certain enzymes, such as the acetylcholinesterase that I mentioned, or in this case, they can be taken up by astrocytes. So that's one of the jobs that astrocytes perform. They will absorb neurotransmitters so that the neurotransmitter doesn't continue to exert its effect on the postsynaptic neuron. Astrocytes also secrete nerve growth factors. Remember, I told you that if an axon is damaged, it can grow back. Well, there are certain growth factors that are released by astrocytes that promote the regeneration of a damaged axon. So if the nerve cell body is unharmed, but the axon has been severed or damaged in some way, it can grow back, but these growth factors are necessary for that to happen. Okay, the oligodendrocytes I mentioned on the first slide, these form the myelin sheath around the axons in the brain and spinal cord. Now that function is carried out by Schwann cells in the peripheral nervous system, but in the central nervous system, oligodendrocytes form the myelin sheath. Myelin is a fatty substance. In order to have myelinated axons, then there has to be fat in the diet. So it is not a good idea to deprive a developing child of fat in the diet because myelin is made from that fat, and the myelination of axons takes place in the growing years. You may have noticed that people get smarter with age. They get better. They can run faster. They can play music better than a child can. Well, the child doesn't form new neurons. We already established that. Well, one of the things that is happening in those formative years is that those axons are being myelinated and they conduct signals faster when they are myelinated. Myelin is made from fat. So if the child doesn't have sufficient fat in the diet, they may not myelinate their nervous tissue properly. So just something to tell little sister about there. Okay, the ependymal cells, those are the little cuboidal epithelial cells that are present in the ventricles of the brain, and they make the cerebrospinal fluid. Well, that really isn't true because cerebrospinal fluid is largely blood plasma. The ependymal cells process the blood plasma such that it is of a different chemical makeup than just straight blood plasma, and so we call it cerebrospinal fluid. The function of the microglia I have already talked about. Let me say one thing before I go beyond that to the peripheral nervous system. These are not neurons. These are glial cells. They do undergo mitosis. Cells that undergo mitosis have a cell cycle that they go through. I hope that you remember this. Cells that go through a cell cycle come up against checkpoints. Well, that wouldn't apply to neurons. Neurons don't go through a cell cycle because they're not going to undergo mitosis. Well, the glial cells do undergo mitosis. So they do go through a cell cycle. So they do come up on checkpoints. Well, those checkpoints are there to inspect the damaged DNA. If the DNA is damaged and it can bypass the checkpoint, then you have cancer. So these are called gliomas. So if a person has a brain tumor, chances are it's a glioma. It's very rare for there to be such a thing as a neuroma, a tumor of nerve cells. Nerve cells don't undergo mitosis, so they're not likely to become cancerous. Well, that is not true of the glial cells. They do undergo mitosis, they do go through a cell cycle, they do become cancerous, and so most times when a person has a brain tumor, it is an astrocytoma or an oligodendrocytoma, or just generally called a glioma, a tumor of glial cells. Okay, well that says that. So down to the last two, the Schwann cells. These are the myelin makers in the peripheral nervous system. So what is the peripheral nervous system? Well, it's anything that isn't central. Well, what is central? The brain and the spinal cord is the central nervous system. So then the peripheral nervous system includes the 12 pairs of cranial nerves. Those, yeah, they come right off the brain, but they are peripheral nerves. The 31 pairs of spinal nerves and all of the nerves that branch from them. So those are the peripheral nervous system. The only thing that is central is the brain and the spinal cord itself. So in the peripheral nervous system, that would include the cranial nerves and the what did I just call those other guys? The 31 pairs of spinal nerves and all the branches from them. Well, if they are myelinated, then that myelin sheath is the Schwann cells that wrap around them. Okay, the satellite cells regulating the chemical environment around the neurons in the peripheral nervous system. Uh, Not a lot is known about satellite cells other than what is stated here, that they seem to perform some regulatory function that keeps the peripheral nervous system acting right. Okay, so that's all I've got for this. I still have a lot to say about neurotransmitters, but I will put that on another slide and have it available for you later.